Hello everyone, thank you for joining me in this presentation. My name is Sylvia Mlemba and I'm going to discuss with you the concept of One Health. Why I want us to discuss the concept of One Health, this is because as human beings we don't live in isolation. We live interconnected together with environments, animals, plants, and we live within the environment. That's why I wanted us to discuss this issue. So our learning objectives will be understanding the concept of One Health, why One Health, and the examples of One Health in action, and then I will take you through a planned case study and then to the conclusions. The One Health approach, this is an interconnectedness of human, animal, environmental, and plants. The connection of all these fields is just for one purpose, that is tackling the global health challenges. As you can see on the picture that I have shared with you, that at one point we are all going to meet. That's human, environment, animals. There is a point that we are going to meet. And the concept of One Health needs the collaboration across disciplines such as human medicine, veterinary sciences, plant sciences, ecology, and the environmental sciences. Again, the importance of the collaboration of all these disciplines is because we need to promote a comprehensive understanding of health and develop effective intervention. The concept of One Health is not new. It has existed for at least 200 years, but it was under different names. The first one was One Medicine, and then it came to One World, and finally we are on One Health. The concept of One Health acknowledged that diseases can pass between humans and animals, and that environmental factors play a significant role in disease emergence and transmissions. So what are the importance of One Health? The importance of One Health, one of it is the zoonotic disease prevention. What are zoonotic diseases? These are the infectious diseases that can be transmitted between animals and humans. Just to mention a few of them, we have Ebola, SARS, avian influenza, and COVID-19. One Health approach helps us in prevention of these zoonotic diseases by identification and control of zoonotic disease reservoirs and vectors. So the reservoir refers to a place or organism like animals, humans, soil, or water that harbors and maintains an infectious pathogen like bats, which are the reservoirs of Ebola virus, or we have water, which is a reservoir of cholera. In other ways, we can call these reservoirs like a home, somewhere the infectious pathogens reside. And for a vector, this is an organism that transmits the pathogen from a reservoir to a host, or from one point to another point. For example of vectors, we have flies, we have the triatomine bugs, and etc. Also, the One Health approach helps in surveillance systems that integrate data from different fields, sources that facilitate early detection and the response of these zoonotic diseases. Another importance of One Health is on antimicrobial resistance control. As all of us we know, antimicrobial resistance now is a crisis all over the world. Everyone will hear cries of antimicrobial resistance. And this is because antimicrobial resistance affects both human and animal health, and it is caused by the overuse and misuse of antibiotics in medicine and agriculture. So the resistance pathogens from which have been caused by the antimicrobial resistance, they spread across humans, animals, and the environments. So generally, antimicrobial resistance undermines the treatment effectiveness and increases the mortality. So the approach of One Health, it helps in reducing the antimicrobial resistance through the promotion of responsible antibiotic use in humans and animals. Also, it helps in strengthening the integrated surveillance system, and it supports research and innovations on resistance and the development of coordinated policies across sectors. Also, we have the importance of One Health approach on environmental protection. So, as all of us we know, once we have environmental degradation, means we will have the ecosystem deterioration, and an ecosystem imbalance will alter pathogens ecology, host susceptibility, and vector biology. With deforestation, means we'll have wildlife stress, and then this will take us to zoonotic spillovers. With water pollution, we have pathogens and toxins, which we can bring diarrhea diseases and poisoning. And then with air pollution, we will have the particulate exposure, which can bring respiratory diseases, cardiac diseases, sometimes skin irritations, and eyes. And then with soil degradation, we will have poor crops, which will result in malnutrition and food insecurities. And climate change will expand the vector habitats, which can cause malaria and dengue transmission. So how the approach of One Health helps us in protecting our environment? This is through the promotion of ecosystem restorations, which reduces zoonotic emergency risks, and then sustainable agriculture, which protects soil, food, and livestock health. Also in pollution control, which lowers chronic disease burden, and climate mitigation, this limits vector expansion and extreme events. Also environmental conservation, which is a long-term public health infrastructure. Generally, the health ecosystem acts as a biological buffer system against diseases. Also, One Health approach helps in ensuring food safety and security. Food safety, this is the preventing contamination of food by biological, chemical, or physical hazards. There are different points of food chain risks, which are the zoonotic pathogens such as Salmonella and E. coli. We have drug resistant pathogens that enters our food through animal products, and then environmental contaminants such as pesticides and heavy metals. So what are the critical control points for these risks so that we have food safety? This is through safe handling of livestock, hygienic slaughter and processing, and then proper storage and refrigeration. So I tried to put a schematic presentation of steps to food safety, which is the first one is clean. So at this stage, it's either you clean the food itself if it needs to be cleaned, or if it is anything that you are supposed to eat, you have to clean your hands before using that food. And then we have to separate the ready to eat food with the uncooked ones or the raw food. And then at the cook stage, it's how you cook. If it's something needs to be properly cooked, or you have to cook it properly. And then the last one is chilling. This is the storage stage. How you store your food matters. We have food security. This exists when all people have physical, social, and economical access to sufficient, safe, and nutritious food at all times. This is according to Food Agriculture Organizations. The key dimensions for food security is availability. This means adequate food supply. And then we have access, which means economic and physical access. And then utilization means the nutritional value and safety. And then stability. This is resilience over the time. It's like that you have food all over the time or throughout the year. We have the threats to food security, which are the climate change and extreme weather, the crop pests and plant diseases, which will result in low production, and then reduced depression, such as on water and soil fertility, resource depression, and then conflict and economic instability. So on these conflicts and economic instability, when people, they are not settled, they are running here and there, they cannot do anything like farming, so we will not have anything to harvest. So how One Health helps for us to have food safety and security? This is through health ecosystem, which supports crop and livestock production, through reduced disease burdens to improve productivity, and sustainable practices to protect land and water resources. And then the global goal of United Nations is to achieve zero hunger by 2030. The following are the examples of One Health in action. We have the vaccination programs. This protects both human and animals, and then it reduces the cross-species disease transmission. Another one is the early warning systems. For example, the global early warning system, which monitors animal populations for disease threats. It integrates human and animal health data, which finally enables us on early detection and rapid response. Another action is collaborative research. Nowadays, people are being advised to collaborate with people from different fields. The collaborative research across medicine, veterinary, and ecology, this supports evidence-based prevention and strategies. We have policy development, and the policy integrating human, animal, and environmental health improves long-term health outcomes. Another example is bioremediation. This is the use of living organisms such as bacteria, fungi, and plants to remove or neutralize pollutants from the contaminated areas. These organisms, they break down pollutants into less toxic or harmless form. You can see in the diagram that I tried to present, the left side is the one that needs to be bioremediated. In the polluted one, you can check there's no life. We have a lot of antibiotics on the ground, and at the top we have dead plants. There's no life at all. But on the other side, we have life even at the underground. We have live plants. So that is after bioremediation. So you can see the importance of bioremediation. So this is a video that tries to summarize what I was speaking to, summarizing all the concept of One Health.
19, Mpox, and Ebola, as well as other zoonotic diseases like rabies, some vector-borne diseases such as yellow fever, and other threats like antimicrobial resistance and foodborne diseases, all illustrate the strong relationship between the health of humans, animals, plants, and the environment we share. For instance, around 75% of all emerging infectious human diseases originate in animals. Human activities, including irresponsible land use and deforestation, result in people and domestic animals encroaching into wild areas. This increases the risk of new or re-emerging pathogens spilling over between wildlife, domestic animals, and humans. Many other factors can affect the distribution of diseases. The climate crisis has led to unpredictable and extreme weather events like floods, droughts, and wildfires, with a huge impact on animal, human, and plant populations, and the environment itself. Similarly, biodiversity loss undermines the ecosystem's ability to mitigate the spread of infectious diseases across species. Collaboration across sectors and disciplines through a One Health approach is a vital need for addressing the complex health challenges facing our society. No one organization can do it alone. Therefore, the Food and Agriculture Organization of the United Nations, the UN Environment Program, the World Health Organization, and the World Organization for Animal Health have come together as a quadripartite. They have launched the One Health Joint Plan of Action to integrate and coordinate strategies and actions across the human, animal, plants, and environmental sectors. This new plan focuses on supporting and scaling up countries' capacities in 6 main areas: health systems, zoonotic diseases, neglected tropical and vector-borne diseases, food safety, antimicrobial resistance, and the environment. It promotes the health of humans, animals, plants, and the environment by providing guidance and tools to coordinate multi-sectoral approaches to prevent and manage health risks at the human, animal, plant, environment interface. There is only one planet and one health. Together, we can build a safer, healthier, fairer, and sustainable world for all. COVID-19, Mpox, and Ebola, as well as other zoonotic diseases like rabies, some vector-borne diseases such as yellow fever, and
So now I'm going to take you through a case of plants in the concept of One Health. This is my master's research thesis, which was on characterizations of common bean resistance to major folio diseases in Tanzania. Why I decided to share this with you as I'm talking on the concept of One Health, as you can see on the title, was for characterization of common beans resistance to major folio diseases in Tanzania, and the aim was to check which germlines, like which lines have multiple Gs of resistance, so as to reduce the use of pesticides in the field. So common beans are the most important grain legume for direct human consumption worldwide, and they are being consumed into different ways. They can be consumed as leaves or the young ones, and then the dry beans. Tanzania, where this research was carried on, is in the first one in the rank of common bean producers in Africa. And I was trying to check on the United States, on the Food and Agriculture Organization data. United States is on the 6th rank. This is according to 2024 Food and Agriculture Organization data. Here in the US, we have some areas which produce common beans, mostly being produced in North Dakota, and then Michigan, Minnesota, and Nebraska. These are the top 10 global producers of common bean, the first one being India, and then we have other different countries, as you can see the way I mentioned, from the first one, which is India, to the last one, to the 10th one, which is Argentina. The problem statement for and justification for doing this study, it's because common beans are mostly being produced by small-scale farmers, and these small-scale farmers, they depend on seeds which they just store themselves yearly. So it's like when they grow seeds maybe this term and they harvest, they will take a portion of those seeds and keep for next season. And these seeds, they are susceptible to diseases, and in the field we have the problem of concurrence of multiple diseases. So you may find these seeds or the plants, most of the time they are being infected by diseases, and then at the end of the time we have low production. So this brought or necessitates for the combination or combining different genes for multiple disease into a single genotype in order to suit the field condition. So the diagram that I've just tried to share with you is the symptoms of common bacterial blight, CBB, on the leaves, as you can see, and on the pods of the common bean. The overall objective of this study was to contribute to bean breeding efforts by characterizing common beans for multiple disease resistance with two specific objectives. The first one was to characterize those common beans under natural field pressure, and the second one was molecular characterization. So this is the methodology for the first objective, which was identification of common bean lines for multiple disease resistance under natural field condition. So the germplasm that I used on this study was collected from the bean breeding program at Sokoine University of Agriculture, which is in Tanzania, and they were just the F1 lines. So I had to advance them to F3 for at least to stabilize them, and then the F3 generation is the one that I took it to the field. And the experimental design that I used in this experiment is the randomized complete block design, which was in 3 replication, and evaluation of the data was performed by the SEAT scale, which is 1 to 9, whereby 1 to 3 are susceptible lines, and then from 4 to 9, this will be resistance line. And the data was analyzed using the GeneStat software version 15. This is the glimpse of some activities of the work which was conducted. The first one here, you can see 2 people. That was just setting the field trial, and at the middle was during multiplication of the seeds. And the first one here, you can see written Sylvia trial. That is the trial after some times after planting the seeds. This is the example of how the disease severity score for ALS which is the angular leaf spot, were determined based on the observation of the portion of the leaf area that is affected. So the MDSS1 means mean disease severity score of 1. So for a plant to be given a mean disease severity score of 1, it means it is just a small portion of the leaf is affected. You can compare the one with 3, or you can check the one with 5, and you can see large area of the leaf is affected. And then this is the methodology for the second objective, which was molecular characterization of common bean lines with multiple disease resistance. And on this methodology, first of all, I had to collect samples from the field, and then these samples were taken to the lab where DNA extraction was conducted, and then PCR. And when I was doing PCR, I had 4 primers, and each primer was specific for each disease. For common bacterial blight, I used the sub-6 primer, and then the gene that I was trying to identify is the major QTL. For bean common mosaic virus, I used the SW13 primer, and then for angular leaf spot, the G976, for anthracnose, SBB14. And at the other side are the genes that I was trying to check. After PCR, I had to do gel electrophoresis, and then I did gel electrophoresis using 2 different concentrations, 1.2 and 2%, and this is because of the Gs that I was trying to check had different sizes. So for the one that has a small size, I had to use the gel which was somehow concentrated, that was the 2%. And then documentation and scoring was just on the band presence and absence. And this is the table, show the mean disease severity score and the natural disease pressure. And the means within the same column followed by the same letter are not significantly different from each other. So you can check, let's say from this disease anthracnose, you can see all means they have the same letter AA except one which has 1.4 and it has a B on it. This mean was different from other that was 1. You can go check also on this ALS. You have at least different means. We have A, we have B, we have AB, and then we have BC. So this letter, they try to show that these means are different. And this table was the data on the growth and the yield components of common bean genotypes, because we had to check like how many days does plants take to 50% maturing, days to 85% of maturing. Then you have to collect the number of pods, number of seeds per pod, and the 100 seed weight, and then the total yield. And this table tries to show the percentage of positive and negative samples when screened with genes specific for each marker. So the total sample that I was screening was 179. So these are the positive samples, the negative samples, and here are the positive samples. You can see on this major QTL, I had most of the samples were positive. Means most of the plants had the gene for resistance for common bacterial blight. And then this is a trend line chart showing relation between genotypes with multiple disease resistance and number of genes present. Here we have the number of genotypes, and here we have the number of genes. So what I'm trying to tell you here, you can check, for example, here we have 1. So it means at least 20 plants or 25 plants, they had just 1 gene of resistance. And then let's say at 3, you can see here, most of the plants had 3 genes of resistance. Most of the plants had 3 genes of disease resistance. So if most of the plants they had 3 genes of resistance, we can say this program or what we conducted, the breeding for multiple gene resistance, was successful. And this is the paper that was published from that work, and it was published in the African Journal of Biotechnology. And here this link can take you through this paper directory. The conclusions from the case study that I took you: as I said earlier, larger proportions of lines screened have shown to contain multiple disease resistance genes, and they presented a reasonably high yield. This means this improvement of these common beans for multiple disease resistance was successful. The evidence from this study suggests that breeding common beans for multiple disease resistance could be one of the strategies for increasing common bean yields. This, in the One Health approach, it is essential for environmental protection and food security. So the future direction of this work is seed release procedures, then seed multiplication and seed distribution. And this is a diagram that's trying to show a general summary of what I was trying to share with you from the beginning to the end. I see like 60% of human infectious diseases they originate in animals. We have the emergence of antibiotic resistance microorganisms, and when 20% of global animal production losses are due to animal diseases. So from this summary also you can get a lot of information that will try to understand or to make us know the importance of doing everything in the concept of One Health approach. Thank you for listening. Meet you in the next presentation. Any question you can share on the comments. Thank you. Bye.